asking yourself, what is meiosis? Well, meiosis is a process of cell division where a diploid cell, a cell with 2 copies of each chromosome, divides to form 4 haploid cells, or cells with only 1 copy of each chromosome. Now, I know you've probably all heard about the very similar process of mitosis. In order to stray away from any kind of confusion, let's go over some of the key differences between meiosis and mitosis. As you can see from this figure, mitosis and meiosis both begin with diploid parent cells. Remember from the beginning of the video, diploid cells mean they are cells with 2 copies of each chromosome. The parent cells then undergo DNA replication, and mitosis then divides into 2 daughter cells, which are still diploid, meaning they have 2 copies of each chromosome. This is the end of mitosis. Meiosis, on the other hand, divides one more time, producing 4 daughter cells. These daughter cells from meiosis are haploid cells, meaning they only have 1 copy of each chromosome. Now that we've gone through some of the basic terminology regarding meiosis, let's go through each individual step. An important part of meiosis is interphase. Interphase occurs before meiosis begins. However, this is the period when the cell goes through growth and development as well as DNA replication. Prophase 1 is the stage in which homologous chromosomes pair up and find each other. This is also the stage in which crossing over or homologous recombination occurs. Here is a quick video to show you just what crossing over, or otherwise known as homologous recombination, is.
During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity.
During metaphase 1, the centrioles, the green pieces of paper you see on the left and the right, have already moved to opposite sides of the cell. The blue pieces of ribbon you see are called spindle fibers, and these attach to the homologous chromosomes. The homologous chromosomes line up in the center of the cell during metaphase 1. During anaphase 1, the homologous chromosomes are lined up in the center for metaphase 1. What happens next is the chromosomes pull apart from each other like so. The next step is called telophase 1. During telophase 1, the cell splits into 2 different cells. This is now the end of the first part of meiosis. During the second part of meiosis, the 2 cells undergo the same process. However, I will only be showing you what's happening in one cell. Recall from metaphase 1 when the homologous chromosomes were paired up and lined up in the center of the cell. This is a very similar process in metaphase 2, although they are not paired up with a homologous chromosome. The chromosomes each individually line up in the center of the cell. The next step is anaphase 2. Remember back to anaphase 1, this is where the homologous chromosomes separated. In anaphase 2, however, this is where the sister chromatids separate, like so. In the final step of meiosis, the 2 daughter cells each divide, giving us 4 haploid cells, or 4 cells with 1 copy of each chromosome.